Hello everyone and welcome to today's live broadcast, Development of a Mammalian Recombinant Protein Production Suite, presented by Catherine Owczarek, PhD. We're delighted to bring you this educational web seminar sponsored by Gibco, part of Thermo Fisher Scientific. For more information, please visit www.thermofisher.com/protein expression. I'm Judy O'Rourke of LabRoots and I'll be your moderator for today's event. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during this presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please note that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Catherine Owczarek, PhD, Principal Scientist at CSL Limited. Dr. Owczarek is the Director of the Recombinant Protein Expression Group at CSL Limited, located at the Bio21 Institute in Melbourne, Australia. After gaining a PhD at the John Curtin School of Medical Research in Canberra, Catherine completed her postdoctoral studies at the Sir William Dunn School of Pathology in Oxford, the Walter and Eliza Hall Institute in Melbourne, and then was a Senior Research Fellow at the Monash Institute of Medical Research in Melbourne. Since joining CSL Limited in 2004, Catherine has led the CSL Research Group's efforts in the successful development of a program to produce mammalian-derived recombinant proteins using disposable cell culture technology. She's involved in a range of CSL's early-phase drug discovery campaigns where there is a high demand for recombinant proteins. Please join me in welcoming Dr. Owczarek. I will now turn the presentation over to her.
Well, thank you very much, Judy, for your introduction. And also I'd like to thank the organisers for inviting me to speak at this fantastic event. So today I'm going to tell you about the development of a mammalian recombinant protein production suite that me and my colleagues have set up at CSL. So to start off with, I'll just give you a little bit of a background on who we are, who CSL Research and Development is, and then outline the types of protein that we use in research and development for all our preclinical studies. And then I'll go on to discuss how we physically carry out the recombinant protein expression using transient gene expression. And then I'd like to go through a few real-life scenarios because there are many different proteins that we come across in our work, and each one seems to require multiple approaches to get the best possible result that we can. And then finally, some conclusions, and then the kind of things we'd like to do in the future. So to start off with, just a bit of an introduction. So CSL Limited is a global specialty biotherapeutics company. And incidentally, we've just celebrated our 100-year birthday. So we opened doors in 1916, but since then we've become a leading innovator and manufacturer in the US, the plasma protein industry. We have the largest plasma collection network. We're a major provider of the influenza vaccine industry, and we have operations in the US, Europe and Australia with more than 14,000 employees in over 30 countries. So the CSL's global research capability, we're actually located at 4 different sites. So I'm at Bio21. Let's see if I can do these controls. On the top right-hand side, and that's located in Melbourne. And then further down is the site at Marburg in Germany. Then there's also a site in Kankakee in Illinois, and then another site at Bern in Switzerland. So we have a single coordinated project portfolio, which means we know what everyone's doing, and it's all towards many common goals. So we have a research excellence in therapeutic proteins, And following on from that, there are plasma and recombinant manufacturing platforms. So just a bit of a background also on the different CSL businesses. There's three major ones. There's CSL Behring, and that's a global business. There's a Securus, which is mainly the flu vaccine provider, and also there's research and development. CSL Behring develops and manufactures therapies for coagulation disorders, immunological disorders, pulmonary therapies, wound healing and critical care, and it operates one of the largest plasma networks in the world. Research and development, which is really the part I work for, its main mission is to develop new and improved protein-based therapies for treating serious illnesses in the main areas of haemophilia, immunoglobulins, specialty plasma products, and breakthrough medicines. So the CSL R&D strategy is to develop protein-based therapies. And as I previously mentioned, they're breakthrough medicines, immunoglobulins, specialty products, and haemophilia products. And they're designed to meet significant unmet needs, the multiple indications. And one of the main things that we have been doing in the last few years are recombinant monoclonal antibodies. Also, there's been a significant amount of work on recombinant long-lasting analogues of human coagulation factors. And as an example of something that we have taken through from the combined global research effort through to FDA approval is a molecule we used to know as CSL 654. And that's human coagulation factor 9 fused to human serum albumin. And the reason we do that is because human albumin can extend the half-life. And you can see in the graph below the half-life extension in haemophilia B dogs compared to factor 9. Once that was shown, we, in the research group, we generated manufacturing CHO clones. It went through the process of a 500-litre fed batch fermentation. Then it was tried in patients, showing half-life extension in haemophilia B patients compared to factor 9. And now finally, there has been approval by the FDA, and our product is called Idelvion. But a little bit more closer to home, and that's in the group that I run at CSL. All the proteins that we make are preclinical and they're research grade, and we do make a very diverse range of recombinant proteins, and these range from recombinant monoclonal antibodies and their derivatives such as FABs, SCFBs. We do soluble cytokine receptors, we do Fc fusions, cytokines, enzymes, iron-binding proteins, soluble type 2 to 4 proteins, vitamin K-dependent proteins, muteins of the above, and quite often we're stuck with uncharacterized proteins. So the types of features for a recombinant protein is that they need to be secreted so that we can purify them. They need to be correctly folded. They need to have correct post-translational modifications such as N and O glycosylation. They need another post-translational modification called Sialylation. For the coagulation factors, they need to be gamma carboxylated, which is very important in order for biological activity to happen. Other post-translational modifications include fucosylation and pro-peptide processing. They also need low endotoxin levels if we are to use these in animal models. And for all these reasons, we have chosen to go down the route of mammalian expression. So the main way we make recombinant proteins is via transient gene expression, which we find is a really rapid way of generating recombinant proteins. And when I mean rapid, I mean that from an expression construct to making a protein can take a few weeks. And this type of protein expression is very suitable for short-term protein production and for high throughput type expression systems. And in general, we do our routine proteins, early stage monoclonal antibodies and Fabs. And the sort of scale that we do in the research group is from the 5 or 30 ml scale to 10 litre cultures. And we find that it's relatively easy to generate 10 milligrams to 5 grams of protein. And the way a transient transfection works is the DNA is added to the cell, which is taken up by the nucleus, expression in the cytosol, and the protein is secreted. Pretty soon after the cells, after 5 or 6 days, the cells are dead or they stop producing, and we can move on to the next protein. So in the research group at CSL, we tried many transient gene expression systems, and we chose to go down the commercially available route. So we wanted ones that were adapted to high density serum-free suspension culture. They needed to have high transfection efficiencies, relatively high protein titer. And the ones that we tried in the early days, we found the most successful was the FreeStyle 293 expression system. And we've since moved on also to the Expi293 system, which was the next generation of the FreeStyle. And now we have also been beta testers for the ExpiCHO expression system. And you can see our real life examples down the bottom. So I'd just like to tell you just a kind of a scenario of the types of the uses for recombinant proteins for discovering novel human therapeutic antibodies. So when a project enters research, we might need to identify, we might have a target, and then we need to identify the antibody, to search for antibodies that might neutralize or just have different types of functions. And the two ways we do them is by phage display or by hybridoma. Then they go through a high throughput generation of these antibodies, so small amounts are made. Then each of these antibodies is analysed in various ways using cellular assays, flow cytometry, and kinetics to see which is the best group of antibodies. And often we'll get a couple of lead antibodies. Then we often find that with antibodies, they might work, but they might need they need to improve either their affinity or their function, and this will go through an iterative process of affinity maturation. And then when a lead is finally generated, then it becomes suitable for in vivo models. We might do epitope mapping, structural studies, generating of anti-idiotype antibodies, and really at the end of this process, it can move into preclinical product development for generating a manufacturing CHO cell line. So practically, how do we make a recombinant protein using transient gene expression in the lab? So generally, someone has to ask for their protein. And once that's occurred, we go about making a gene expression construct. Then there's the process of making the DNA prep. We need to do a pilot transfection to see how much we need to produce. Then we scale up the transfection, and finally it goes on to our purification colleagues who turn it into the final product for our researchers. Just a few practical things on how we do things in my group, and really the type of culture vessels that we use depends on the volumes required. So sometimes we get away with these little mini bioreactors, and they're from 5 to 10 mils. Often we use e-flasks, and they can be from 30 mils to 1 litre. And in many instances we use wave bioreactors, and they can be anything from 5 litres to 10 litres. And we find that we get relatively good scalability for the smaller volumes from 5 to 1 litre. 5 litres sometimes drops a little bit, but we can compensate for that. One important thing that we do in the lab is to use synthetic genes. And the reason we do this is because you can generally really improve your expression levels. We can remove sequence repeats, codon optimization, we can remove potential splice and polyA sites and RNA secondary structure. And compared to traditional cloning, it's very quick to obtain your cDNA. There's no need for the source of mRNA. The sequences are always accurate, and you get rapid generation of isoforms and mutants. And we use commercially available sources such as GeneArt for doing this. And just here's an example of the types of things you might see when you do your codon and sequence optimization. So we had an instance where we had several Fab fragments that we generated recombinantly. We only had the light chain optimized, and we really didn't recover any intact Fab. So we went back to the books and or the drawing board, and we optimized the heavy and light chain, and we were able to get 30 to 80 mg per litre excuse me, of intact Fab recovered. So in this instance, and in many others, we find that it improved things quite substantially. And recently, we look at ways of improving our protein titer, and often we'll have an antibody which is composed of a heavy and light chain. Since we started working with the ExpiCHOs, we've been investigating how we might improve, get the maximum expression yields that we can out of the CHO system. So what we've been doing is looking at the expression levels of the wild type or non-codon optimized sequence relative to a CHO codon optimized sequence. And we've also been doing the heavy to light chain ratios. So what we find is that if we have too much light chain relative to heavy chain, we might get less optimal protein expression. So when we go from the 2-to-1 light to heavy, it's increasing, and then you go from 1-to-1 light to heavy, and that's when you get your, in this case, your maximal protein expression. And we found, interestingly, that the CHO optimized was and this would have been an ExpiCHOs, would have been the best levels obtained. And a phenomenon we hadn't noticed before was that when we have too much light chain, the cells seem to die quite rapidly, and you get no protein at all. So then I'd like to go on to some real life scenarios that we have, and they're kind of issues that we deal with all the time. And I'm going to tell you about some things that we've done. Sometimes we haven't got the full solution, and I've told you before about the different cell lines that are available to us, the FreeStyle 293s, the Expi293s, and the CHOs. And in my group, we've decided to keep all three because we have the luxury of being able to run 3 cell lines, and we find that we have different proteins that require specific characteristics. So when someone asks me to produce a protein, they might say, look, I need this protein because I need correct biological activity, and it needs very complex post-translational modifications. Someone else might want just a whole swag of his-tagged proteins. Sometimes they're not very well expressed, they aggregate, but they need to do the variants, say for epitope mapping. I might have another researcher who will be doing an in vivo animal model, and they will need to show that in order to get the effect that they want, the protein needs to stay in the animal circulation for sufficient time to have that effect. And quite often, well, not too often, we get the mad, crazy, urgent requests where an animal study or something has been set up, and they really need, the clinical research or the research organisation really needs the antibody. They'll need 10 grams of AmAb, and they'll also really need the isotype control. And we'll have a situation where we really want to accommodate people's requests. So we will choose different cell lines for different purposes. So firstly, I'd like to talk about the mad, crazy, urgent requests where we get 10 grams in a short time, and plus the isotype control antibody. So that equates to 20 grams of mAb. And we're not really set up for manufacturing. We're research grade, so these kind of things in the past would pose a challenge. And the way we will be able to get around this is by using the new ExpiCHO expression system. And we find that we really do need a high producing transient expression system for projects requiring monoclonal antibody quantities and greater than a gram. We tend to avoid doing large volume just because of the manual handling and the pressures it generates for people doing the expression, but also the purification. So we were interested in the ExpiCHO because CHOs are obviously an attractive candidate because you have the potential to have a similar cell line from research to manufacturing. And we were very lucky to be beta testing participants, and we've also done a few in-house optimization experiments. So I'm just going to quickly run through some of the things we've done during that beta testing and following on. So showing here is just a slide showing how we have improved titers overall with the ExpiCHO system. And maybe a few years ago I would have shown the difference between the FreeStyle 293Fs in blue compared to the Expi293Fs. And we used to be very happy with our FreeStyle expressions. With the Expi293 we would get quite a substantial increase. And with the very same panel of monoclonal antibodies, we've shown that overall we can get quite marked improvements in our system. So it's early days for us yet, but we're still very happy with the 1.5 grams or so that we're obtaining with a lot of the antibodies that we're using. So the antibody that I've marked with a star is the one that we used for many experiments. I've just pulled our star antibody, so that was the beta testing antibody. So what we showed, we needed to show that we had scalability from 25 mils to 200 mils, and that was all very good. And we would have done the max titer transfection. And importantly for us, we needed to show that the monoclonal antibody expressed in the ExpiCHO had similar potency to that expressed in the Expi293s, and it was virtually identical. We also needed to show that the monoclonal antibody expressed in these two systems had the same association, dissociation, and affinity for the receptor. And shown here are Beer-core traces showing that both antibodies, both from the Expi293 and the ExpiCHO system, had similar on and off rates and affinities. So we were pretty confident with this antibody, at least, that the antibody produced in both systems was virtually the same. We were also lucky enough to have monoclonal antibodies an analysis system using mass spectrometry. And my colleague Matthias Peltzing was able to take the protein derived from ExpiCHO and Expi293s and show that it had both had the correct light chains with the correct isotopic mass. He was also able to do some glycosylation analysis, and in this case we did observe some difference between the Expi293 and the ExpiCHO. So the ExpiCHO appeared to have less mature end glycosylation. We haven't really determined the significance of this yet, and whether this particular antibody in a manufacturing sense would have exactly the same, but because for our purposes it has the same biological activity and other metrics, we haven't really investigated this too much further. So just in a nutshell, we think that because of the increased volumetric yield in the ExpiCHOs for 2 grams of this particular monoclonal antibody, in the old days we would have been trying to produce in the CHO cultures the FreeStyle CHOSs, and they gave us 40 mg per litre, and we would have had to have done 5 by 10 litre cultures to get this amount of antibody. Moving up to the FreeStyles, that would have been 69 mg per litre, so it would have been 3 10-litre wave cultures. It was happy days for us when Expi293 came along, only 1 5-litre wave culture, but now we feel that with the ExpiCHO we can really reduce our volumes and the amount of work we have to do. So to get our 2 grams, we'll only need 2 1-litre e-flasks. Now moving on to a sort of a trickier problem, and that is making proteins with the correct in vivo half-life. And this is something we've been only recently having to deal with, because monoclonal antibodies have very good half-lives in vivo because they go via the FcRn receptor and they get recycled. Not so the case with several other types of proteins. Now the issue is with these proteins is that they need a modification called sialylation, and that's adding a sialic acid residue to the N-glycosylation site. And this can affect biological activity but most importantly, the in vivo circuitry half-life of glycoproteins. And it's quite well known that mammalian cell lines can aberrantly and variably sialylate recombinant proteins. This tends to be protein specific, and it can be affected by the culture conditions. So we're in our early days of doing this, but I'm going to show you just what can happen with a recombinant protein. And we're moving down the path of glycoengineering, so where we can co-transfect specific enzymes to increase the sialic acid level of these proteins. So shown here is a mass spectrometry analysis showing a plasma-derived protein. And this particular protein has 3 N-glycosylation sites. And you'll see here that the traces show that each N-glycosylation site can be not sialylated. It can have one sialic acid group, and that's the little purple diamond on top of the yellow circles that's stuck at the end of the N-glycosylation site. A lot of them in the plasma-derived protein have 2 sialic acids, and then a lot have 3 sialic acids. However, when we go to a recombinant protein, we find that we're not getting as many sialic acids on there, and that's shown in the top panel, where mostly we have this thing called neutrals, which means it's not sialylated at all. There's a little bit of monosialic acid, a tiny bit of disialic acid, but relative to the plasma-derived, there's hardly any at all. So you can see here in this next slide the consequences of not having sufficient sialylation. So in this graph here, the red circles is the plasma-derived protein, showing the clearance from the circulatory system of a mouse. And you'll see that the plasma-derived protein lasts for 48 hours, whereas the recombinant protein is very rapidly cleared. And our current experiments are working on ways we can improve the sialic acid level of these proteins by either transfecting in sialotransferases or modifying our culture conditions. And maybe next time I'll be able to tell you how we solve this problem. Another option that we are interested in is generating proteins that require complex post-translational modifications for correct biological activity. And one major thing, a type of protein that CSL's interested in is coag factors, and there's many of them, and they all have their own different ways of doing things. And a lot of this, one characteristic of them is they have pro-peptides, and the pro-peptides need to be cleaved for the coag factor to become activated. Another very tricky PTM is called gamma carboxylation, and that's traditionally been difficult to do recombinantly. So this process requires vitamin K, and we also think it actually requires a low expression level. So you can make loads and loads of protein, but you might find that it's not actually active at all. So what we want is quality proteins that there's a small amount, but they're well gamma carboxylated. Another type of PTM, which is a bit tricky depending on the protein, is the cleavage of a precursor molecule into an alpha and beta chain. So sometimes, like antibodies, you have the heavy and the light chain on different constructs. But this way, it's a single mRNA, and they get cleaved by another enzyme. Now, that enzyme may not be in the HEC293 or the CHO that you're using. So we need to work out how we're going to get it to cleave. Just on this cleavage thing, so sometimes with our recombinant coag factors, we don't really care about the biological activity, and shown here is one such case, but we do need to have the pro-peptide cleaved. So down the bottom is the schematic of a pre-pro coagulation factor zymogen, showing in pink the signal sequence, then in red the pro-peptide, and after that is a furin cleavage site. This is a dibasic cleavage enzyme. It seems to be that in the HEC293s, the Expis and the FreeStyles, there is some cleavage going on, because you can see that in the band on the left, in the lane on the left, there's a high molecular weight and a low, but when you transfect in more furin, you get complete disappearance of the high molecular weight band. One important thing that we've had to learn is that really less is more when it comes to biologically active coagulation factors. And we went through a series of experiments where we added in furin, and on the left-hand side is a coag factor, 2 different coag factors, showing in pink the activity when you don't have furin, and then when you add in 10% furin or 30% furin, and showing here that this seems to improve the biological activity of your vitamin K dependent coag factor. And there are other things that were important for these factors. So we found that a combination of things helped. So there's another enzyme that's relatively low in expression in HEC293s, and that's called VCORC1. That's part of the vitamin K cycle, and that helped the activity. But a major thing was to reduce the amount of cDNA. So there's fewer precursors made in the endoplasmic reticulum of these cells, and the fewer precursors means there's much more substrates available to be gamma carboxylated in the cell, and then you end up with more biologically active protein being secreted. Another type of molecule we have, which is not related to coag factors, is the cleavage of a molecule into an alpha and a beta chain. And you can see here again on the left the pre-protein, a big fat blob of unprocessed material, showing that you can purify some of it, but when you try and purify it, you end up with a mixture of unprocessed and processed. And then when you add the protease in, you can get cleavage specifically into the alpha chain and the beta chain. Now this relies on knowing what your protease is. So in this case we were lucky, we knew what it was, but sometimes you don't really know. So you really sometimes need to delve into the literature, years and years back to see what protease is relevant to your particular protein. Another type of problem, not a problem, but we're slowly working on solving it, is when we have many variants of poorly expressed His-tag proteins. Sometimes our His-tag proteins express really well, but as life would have it, when we need to work out the epitope of an antibody, or we need to change the activity of something, it always never expresses properly. And one thing that has been bothering us for many years is the purification of histidine-tagged proteins. So the way our protein purification group does this is to use immobilized metal ion affinity chromatography, or AmAb. And we have a bit of a problem here in that we get higher expression levels of most of our His-tag proteins in the Expi293F cells compared to the FreeStyles, but there's an issue with purifying the proteins from the Expi293 media. And this is where we're investigating this, and we think we're like part of the way to solving it. And we think there's components in the media that interfere with the AmAb. And this shown here, this slide, is just a real-life example of how different proteins often express a lot better in the Expi293s compared to the FreeStyles. So on the left-hand side is a cytokine, and you can see down the bottom there's the S stands for FreeStyle and E stands for Expi, and you didn't get anything at all in the FreeStyle, but you got something in the Expi. And similarly, a different type of transmembrane protein, extracellular domain, way better expression in the Expis. So we've ended up with kind of a bit of a complex way of purifying these His-tag proteins. There's different scenarios. So sometimes there's low expression in both systems, so we need a large culture volume. And in this case, we choose the FreeStyle media and a product called nickel sapphire 6 from GE. It has high capacity, but the nickel can be stripped. if there's a large volume of Expi media. So in this case, we go for FreeStyle. Now, if there's good expression in the Expi media, Expi293 cells, we'll use the Expi293 media. We'll use a small culture volume. And when there's a small culture volume, it's okay. It doesn't seem to strip the column. However, when there's good expression in both systems, but there's large amounts needed, we can either use large amounts of FreeStyle or Expi media, but a different resin will be used. And this is called the nickel sapphirose XL, also from GE. It seems to have a lower capacity, but there's no stripping. So we just need to balance expression levels and how much we need to isolate and the purification method against what's required. So let me just carry on, beg your pardon. So this is, so actually that brings me now to my conclusions. So you can see we have a wide variety of cells and conditions that we use in our recombinant protein, and we find that our best expression levels and functionality of recombinant proteins can be obtained by codon optimization, choosing the appropriate cell transfection system. And you need to be aware of the molecules involved in the post-translational modifications. And a lot of our work involves optimizing and changing the processes. So there's a lot of process development depending on the protein that is required. So the conclusion that we have is that we've decided to keep a suite of transfection systems for generating these complex proteins. So we have our ExpiCHOs when we need high protein titers. We will use the Expi293s or the FreeStyles for correct glycosylation. Depending on the expression levels, we might use the Expi293s or the FreeStyles for a his-tag protein. And I think it will depend on the protein involved, whether we need to use ExpiCHOs, Expi293s or FreeStyles for the, the correct PTMs. So in the future, we're hoping to increase the integration of the ExpiCHO system into our routine protein production. So a lot of our high throughput mAbs, we're currently using Expi293s. We'll be working towards putting the ExpiCHOs into that system. We're getting into transcriptomics so that we can understand what is there and what isn't there to help us with our post-translational modifications, and the new phenomenon of genome editing, CRISPR-Cas. We might be able to take out inhibitors, add things in. So this is all sorts of things that we really love to get under our belt. And finally, acknowledgements. So it's all the members of the CSL research group. In particular, I'd like to thank my group, Matt Hardy, Therese Lynch, Jess Petrarca, Susan Wan and Essen Donaldson for all their hard work and what they put in. All this data is theirs. Protein Technologies for all the help with purifications, analytical biochemistry, and antibody technologies. So thank you for listening to me, and I'd be happy to answer any questions.
Thank you, Dr. Owczarek, for that informative presentation. It is time for Q&A. If you have a question you'd like to ask Dr. Owczarek, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many questions as we have time for. Let's get started. So our first question is, what sort of proteins have you encountered that are difficult to express?
Do I answer? Okay, hi. We do encounter quite a few difficult to express proteins. Some things that have defeated me are the kind of membrane proteins that seem to resist secretion. So they're the type 2 proteins where the N-terminus isn't facing out, but this isn't always the case. Sometimes we've had really tricky bacterial proteins to express, and they tend to aggregate a lot. So mostly I try not to admit defeat, but I have been defeated occasionally. But generally we find that with all our techniques, there's some way that we can get around doing this. But mostly the problems are when they're from other species, I think, non-mammalian species that are the more difficult ones, and ones that aren't designed to be secreted into the extracellular medium. So these ones generally are the worst, and occasionally we'll have to go back and say, well, really, is this kind of the target that we want to look at if it's never been expressed on the cell surface? So yeah, those kind of things. Okay, any more questions?
Indeed. What steps do you take when you encounter these proteins?
Okay, so when we get a difficult protein, and I can use an example where it's a protein from a bacteria, and on its own it either doesn't express or it aggregates. One trick we have up our sleeve is to use fusion proteins. So we'll often put something onto human serum albumin or the Fc part of an immunoglobulin. And we can use the secretory power of albumin because it's one of the most abundantly expressed proteins in plasma, and all of the immunoglobulin, and this seems to stabilize the protein. So we might still get a few aggregates, but generally we find that we can have this. And you don't have to put up with the albumin stuck on there. You are able to put, or the Fc, you can put on a protease cleavage site such as thrombin and purify away the albumin using various affinity techniques. So that is one trick that we've developed over the years to get around these difficult proteins.
How do you overcome issues such as aggregation of proteins?
Yeah, aggregation can be a big problem with proteins, and it's really not predictable. Often we'll find that with some antibodies, we'll be getting, wondrous expression levels, and then we hand it on to our purification colleagues, and they'll say, well, sorry, it was all, once they do their size exclusion analysis, it was all high molecular weight. So the things we do to try and avoid that, we'll take a time course, and we'll take samples, at 4, 5, and 6 days post-transfection, and we'll see whether the aggregation is happening early on, and then we can try and reduce that amount. And in some cases, we've been able to solve it by stopping the transfection early and before the proteins had time to aggregate. So in those situations, we've had success. So I know my advice would be to just look at each protein individually, take a time course. Sometimes reducing the temperature can help, but yeah, it's a very big problem.
We're almost out of time. This will have to be our last question. How do you go about increasing protein yield?
Yeah, it's a good question. So prior to the ExpiCHO system, which really has improved the yields quite a lot, especially in antibodies, we would do a lot of process control. So with the Expi293s, we found that the addition of additives such as leucotone lupin really bumped up the expression levels, and it seemed to stabilize the cells and make them survive longer, but also importantly produce a lot more protein. So that was an important addition. Occasionally we'd find that the addition of sodium butyrate helped, with the proviso that it didn't change the biological activity of any protein, and this would somehow put the cells into a kind of semi-senescent state. So instead of growing a lot, they produce more protein, and when that was combined with lupin, that really improved the expression levels. As I mentioned during my talk, we would optimize the heavy to light chain ratios. Sometimes we need to change the signal peptide. Sometimes the endogenous signal peptide doesn't really help. By putting a different one on, somehow the expression levels, tripled. So yes, sometimes it's a combination of things.
I would like to once again thank Dr. Owczarek for her presentation. Do you have any final comments?
Oh, look, it's been a pleasure giving the talk here, and really all this work, hasn't been possible without a lot of people, analytic and protein technologies and the expression group. So yeah, it's been a real pleasure to be here, and you know, we have a good time doing all the work we do at CSL, so I can highly recommend it. So thank you.
Thanks again. I would also like to thank Gibco, part of Thermo Fisher Scientific, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May 4th, 2017. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. We invite you to share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you soon. Goodbye.